Hello, myself Priyanka Rao. A very good morning to all of you. Today's Today I want to explain about T cells. T cells originated in bone marrow, but they are mature in thymus. During maturation, they express an antigen binding T cells receptors. They don't produce antibodies. They perform effective functions when antigen presenting cells bring antigen into secondary lymphoid organs. So today, first of all, we explain about T cells receptors. It consists of heterodimers, which means T cells receptors are heterodimer. They consist two transmembrane glycoprotein chains called alpha chain and beta chain. Alpha chain similar to light chain of B cell, where beta chain, which is similar to the heavy chain of B cell receptor. The transmembrane segments of alpha consist two positive charged amino acids and beta chain have one. This look like that. Two uh, two positively charged amino acids. Here beta chain consists one positive charged amino acids. In 90% of human, more than 90% of human T cells, the T cell receptor consists of an heavy chain and a light chain and a beta chain. Whereas in less than 10% of T cells, T cell receptor consists of gamma and delta chains. Hence, there are two broad category of T cells based on the nature of T cell receptor, which means that we can divide the T cell receptors on the basis of the different chains in two categories: alpha beta T cells and gamma and delta T cells. First of all, we explain gamma and delta T cells. Gamma and delta T cells mainly are lower in number, but during infection their number is increased. In human, alpha and delta T cell consist 0.5 to 1.0% of T cells, where delta T cells, delta, gamma and delta T cells recognize a broad range of antigen. They are very much in size and composition and structure, which uh, which I can explain uh, here in first point. In key, delta and gamma T cells are lower in number, but during infection, their number is increases. So they can recognize a broad range of antigens. They are very rich in, very much in size and composition and structure. They recognize protein and non-protein antigen that are neither proceed or not presented by a classical MHC molecule. So I hope we all understood about delta and gamma T cells. Here, co-receptor molecules. T cells co-receptor molecule. The mature T cells also express the other two co-receptors, Cd4 and Cd8, which are very familiar to all of you. Cd4 is a single chain molecule which is called as monomer, where Cd8 it has two chain molecule called as dimer. CD4 it's bind selectively to the MHC second presenting on APC antigen presenting cell, where CD8 it binds selectively to the MHC first present on antigen presenting cells. It's very important points. And CD4 or CD8, CD4 are monomer where CD8 is dimer, and most important point is that CD4 binds the MHC second present on antigen presenting cell, where CD8s are bind selectively to the MHC first presenting on antigen presenting cells. Most important points these are: the intracellular segment of these co-receptors CD4 or CD8 are linked to enzyme called protein tyrosine kinase and help in the process of T cell receptors. Here the intracellular segments of these co-receptors linked to a enzyme, एक enzyme से linked होती है, which is called as protein tyrosine kinase, and these are help in the process of T cell activation. They are involved in the signal transduction process. Here we see that CD4 or CD8. Here CD4 are the monomer. CD4 are the monomer because it consists only a single chain. Here CD8 are the dimer, which it consists two alpha and beta chains. Here intracellular segments. These are the intracellular segments. These are linked to an enzyme tyrosine kinase, protein tyrosine kinase, which help in the process of cell activation. Here, T cell receptor complex. T cells also express a molecule called as CD3. The CD3 is presented in all T cells. Hence, it is a cell surface marker of T cells. Here, CD3 are the molecules that are present in all T cells. Here, it is a cell surface marker of T cell. Alpha chain plus beta chain plus CD3 together called as T cell receptors complex. Anyone on which are asking you about T cell receptor complex, you can say simply that alpha chain beta chain plus CD3 molecule together called T cell receptor complex. Here CD3 is a chaperone and help in the transportation of uh, T cell receptor molecule to the cell surface. CD3, which are present an extra molecule with alpha or beta chain, which is called a chaperone, they help in the transportation of T cell receptor molecules to the cell surface. CD3 does not bind to the antigen. Here, CD3 is not bind to the antigen. They only help to transportation of T cell receptor molecule to the cell surface. CD3 chain contains ITAM, immunoreceptor tyrosine based activation modify sequence, which functions in signal transduction. Here CD3 molecule contains ITAM, which is whose function is signal transduction. Here CD3 is a complex of CD3 is a complex of 5 polypeptides that associated to form 3 dimers, which is are that gamma and mu, delta and mu, phi, phi. We can see here. These items are presented as the intracellular segments which reaches to the nucleus for signal transductions. These are seen in this diagram T cell receptor CD3 complex. So I think we all are understood about CD3 molecule. So anyone ask about CD3 or T cell receptor complex, we can easily say or we can easily explain about T cell receptor complex. T cell receptor complex are alpha, simply alpha beta chain plus CD3 together called as T cell receptor. These CD3 molecule is a chaperone which help in the transportation of T cell receptor molecule to the cell surface. Here, and CD3 does not binds to the antigens. It also contains ITAM which are presented on intracellular segments, and they are connected to a nucleus and help in signal transduction. So next topic is on the basis of presence of CD4 and CD8, the T cells are. Here we can we divided the co-receptor T cell co-receptor molecules. We divided the mature T cells also express other two co-receptor which is called CD4 or CD8. And here on the basis of these CD4 and CD8, the T cells are divided into T helper cells and T cytotoxic cells. Here I explain that CD4 are the monomer and CD8 are the dimer. T helper cells carry CD4 membrane glycoprotein, where T cytotoxic cells carries CD8 membrane glycoprotein. CD4 provide protection both no sorry T helper cells provide protection both extracellular and intracellular antigen pathogens. Here T cytotoxic cells provide protection against intracellular antigen and pathogen. It is most important point, guys. Here a T helper cells function is to protect both extracellular and intracellular antigen pathogens, and T cytotoxic cells provide protection against intracellular antigen and pathogens. T helper cells stimulate B cells to make antigen and activates macrophages T cytotoxic cells. They can differentiate into effector T cells, subsets T helper 1, 2, 17, FH. Here, T cytotoxic cells, they does not make antibodies or produce antibodies. They kill the infectious cell before the antigen can proliferate and accept from the infected cell to infecting neighboring cells. Thank you so much, guys.